Thank you all for coming. It's my pleasure to introduce Luke Busta. He came all the way from Duluth, Minnesota, where there's not even a hint of spring. The ground's not frozen, so we will say that. But Luke got his PhD in chemistry from the University of British Columbia. He then got an NSF Plant Genome postdoctoral fellowship with Ed Cahoon in Nebraska. And since 2020, he's been an assistant professor in the Department of Chemistry and Biochemistry at University of Minnesota, Duluth. What's exciting is Luke's the recipient of the 2026 Biochemical Society of North America Arthur Nesch Young Investigator Award. So he's obviously a rising star in plant specialized metabolism. He's also the 2025 recipient of the Comparative Scholar Award from the Research Corporation for Science Advancement. What's important in terms of what his research is, a little bit about some grants he has, projects he has. He's working on sorghum grain, which is a source of natural waxes. And then also the reason I invited, not just he works on plant specialized metabolism, but that he's doing a lot of artificial intelligence work in plant specialized metabolism. And for those of you who are not plant people, the biggest driver of chemical diversity in plants, of which, there's 400, 500,000 chemicals, is gene duplication, genome duplication, and trying to crack that nut will require artificial intelligence. And so I think this is one of the first people starting to use AI to try to help us mine the historical nature that we have in articles about what metabolites plants make and how to then use that to make predictions about metabolism. So Luke?
Thanks, Robin. It's still good morning. Yeah, good morning, everybody. It's very nice to have a seminar in the morning. Our seminar is on Friday afternoons, which is a good time. But thank you for the invitation. I'm very happy to be here. As Robin said, when I left Duluth, it was foggy and 30 and the ground was still partially frozen, and what wasn't frozen was mud. And I told my people I was going somewhere green and warm and wonderful here. I'm staying for the weekend. I'm going to go up in the mountains and enjoy your lovely landscape. So thank you for sharing it with me. As Robin mentioned, I'm going to talk about language models today, or artificial intelligence, although I'm also going to talk about plants. And I think that's where we're going to get started, if this thing will. Very good. All right. So plants, as you I'm sure have heard, I know that the people in this audience are broad and study more than just plants. So the plant part's going to be quick. Don't worry. Well, sorry, quick. The plants, of course, are masters of chemical biosynthesis, making a wide variety of small molecules and big molecules. Some examples, of course, are drug molecules like this one here, artemisinin, or some sort of larger molecules in quotes maybe, like cotton. If you're wearing a cotton shirt, this is, of course, a plant product. The one that I'm going to talk about briefly here at the beginning is this structure here. This is a polyester matrix that is called the cuticle, and it's found on the surface of plants. It prevents water from leaking out of the surface of the plant. It's really remarkable, actually. If we think about the surface areas of plants, I can see a tree out there. If I look at all the leaves on the tree, and maybe these are the leaves, if you think about all the leaves in the surface area that they comprise, it's actually really remarkably high. And of course, this is to have high areas for photosynthesis and so forth, but the cost of that is a high area for transpirational water loss. If we try to approximate the surface area of plants across the globe, maybe we get something like 10 to the 9th kilometers squared, which is like on the scale of the surface area of the oceans, at least according to some estimates here. But really what this means is that plant surfaces are one of the largest interfaces between the biosphere and the atmosphere, particularly with regard to the movement of water. That's important. This is emphasized by the amount of freshwater use that goes into irrigating crops. And so these barriers at the surface of the plant sort of see a fair amount of that water. All right, so the first part of my talk here, I'm going to briefly talk about plant surface chemistry. This is because it's sort of where a lot of our work began in my lab, and it leads to a couple other topics. One is the phylogenetic distribution of plant chemicals, as Robin mentioned. And of course, that took us into this space of systematizing research data with language models. And so I'll talk about that as well. I'll try to move through these two relatively quickly because I think there's interest in this area. So I apologize if it moves quickly, but the goal behind that is to get to some nice examples and information that I have here for you in this third topic. Briefly, plant surfaces, you can see here they're super hydrophobic. This is water beating up on the surface of the plant. The superhydrophobic nature is really caused by what are called wax molecules that are often present as crystals on the surface. There are two processes required to get those molecules there. One is, of course, the synthesis of the molecules that make up the surface coating, and then the export of those molecules to the surface itself. And I'm going to come back to that distinction a little bit later, but both are required, of course. We wanted to better understand the chemistry of these surfaces because they see so much of that water movement that I described a moment ago. There are a variety of ways to do this that we've been exploring, including working with herbarium specimens. But one of the things that we did a few years ago was to work with citizen scientists to examine really thick wax deposits on the surface of plants. Here you can see wax is present as this kind of white coating and removed where the green is exposed. We worked with a variety of citizen scientists from K through 12 students through college students. We had a variety of participants all over the country. We mailed them little cotton swabs with which they could collect the samples and return them to our lab. And one of my first graduate students, Dean, analyzed these samples. This is a pruned phylogenetic tree showing a variety of the orders, plant orders from which we received samples. We had 228 from 26 different orders that spanned dicots, monocots, and gymnosperms, which are major groups of plants. Here are some of the chemical compounds that we found in those samples. They belong to two categories: aliphatic compounds, which are very common across most plant species as far as their surface chemistry is concerned, and alicyclics. These are terpenoid compounds, which are a bit more rare. And you can see that some compound classes were found in a high number of species, like these fatty acids were found in 30 of the 78, while some of the terpenoid compounds were found in as few as a single species, meaning that these are generally lineage-specific metabolites found in only certain plants. And this is an important concept, the distinction between chemicals that are broadly found across the plant kingdom versus ones that are found in just restricted lineages. We became intrigued by that. And of course, there are statistical methods for detecting that type of phenomenon, and I'll explain it conceptually here. So here's that tree. This is at the genus level. And here you can see some of the different chemical compounds that we found across these genera. These three groups are groups that were found broadly distributed across the whole tree. You, of course, don't need to look at some of the little details here. The point is that the green, purple, and red bars are widely distributed across the tree, and that this is in contrast to certain other classes that we find. These orange bars here are the triterpenoids I mentioned earlier, as well as some compounds called secondary alcohols that are also found clustered together in small groups as opposed to distributed broadly. We're very interested in these patterns because they can, essentially what they signal is that something interesting happened at this point in the phylogeny that led to a unique chemical phenomenon. And the one that we picked up using the citizen science study was this one here. Something happening at the base of this region of the phylogeny, really at the base of the lineage Kalanchoe, and related species that led to these high abundances of triterpenoids occurring on their surfaces. All right, so this sort of is what this is like one aspect of my lab. We work on the surface chemistry of plants, and as Robin mentioned, we have a USDA project in that area looking into industrial uses of those chemistries. But what I want to talk about now are these distributions that I alluded to just a moment ago. I explained how we found this unique lineage that had these triterpenoids in it, and we decided to follow up on that study. We collected 22 different species from that lineage. A couple of undergraduates in my lab, Alexis and Nicole, very diligently did some work together with Kylie and Noah, two chemistry students in my lab. They looked at the abundance of waxes. Again, this is that special trait that we were looking at, the abundance of the waxes. They looked at the abundance of waxes across all these species. And not only the abundance, but they also looked at the different types of chemicals that were present. I mentioned earlier that the ones that the citizen science screen picked up were terpenoids, but that's not all that's there. They found that, yes, some species in this group indeed have high abundances of triterpenoids, but other species in the group have sort of a broad distribution of compounds present on their surfaces. And still other species have mainly other types of compounds like this alkane here. But what it showed is that within this lineage, there is one particular group that seems to have this unique chemistry that we picked up in the citizen science screen. Nicole extracted DNA from the various species that we had collected and built a phylogeny of these. And the goal here was to be able to put the chemical data that we had collected into the context of a higher quality phylogeny. And so we did that. Here you can see the phylogeny that we prepared, as well as the chemical data mapped onto it. This plot here shows the abundance of those triterpenoid compounds in orange versus the other compounds in green that are these kind of long chain compounds that are more common and found in almost all plant lineages. Of course, one sticks out as not only having high abundances, or excuse me, high purity of triterpenoids, but also high abundances of triterpenoids. This species here, Kalanchoe thriciflora. And we decided to focus in on this one more because what it means is that if we're going to find the genetic basis for this unique chemistry that was picked up in the citizen science screen, this particular species might be a good one to study because it exhibits that quality to a high degree. All right. At this point here, we had some sort of questions, right? So we've got a high abundance of these triterpenoids. How did this come to be? It's a lineage specific phenomenon. And something funny here occurred to us. These triterpenoid compounds, the ability to synthesize them, even though we see them on the surface as this lineage-specific phenomenon, making triterpenoids in general is something that's common to almost all plants. There are triterpenoid-derived hormones that are found broadly across the plant kingdom. And so this became a point of interest for us, and it gave us an idea. We thought, well, we know that there are these two modes that are required to get compounds to the surface of the plant. The synthesis, which as I just told you, seems to be relatively common across plant species. And the second phase is the export. That's also required. And that made us think, well, maybe what's unique about this lineage and the reason we picked it up on our screen has more to do with this export phenomenon rather than with this synthesis phenomenon, which seems to be pretty widely distributed. And so we began to look into the ABCG transporters, which are well-known transporters involved in the transport of waxes, though nobody has really looked at whether they can transport triterpenoid compounds in the context of plant surfaces before. So this is work that we undertook. With these species, or I should say with all plant species, the surface chemicals are synthesized in the epidermis, as you might imagine. And on these species, you can peel the epidermis off. So we isolated epidermal tissue, extracted RNA, assembled transcriptomes, with high sort of Busta scores, so reasonable coverage here. And we began to look for ABCG transport genes that might be involved in moving these triterpenoids to the surface of the plant. And this is where we kind of came across a bit of a stumbling block because the ABC transport family is massive, and really there is a particular architecture of these that's challenging. They can exist in full-length forms, like is shown in these two diagrams here. And in those forms, they're capable of transport as a single peptide. They can also exist in these half transporter forms that require dimerization to work. And so if we're trying to compare previously characterized transporters against these, it becomes tricky when using multiple sequence alignment because it's unclear if we're supposed to align this portion with this portion here, or should we be aligning this half transporter with this portion here? What about this half transporter's partner? Does it have multiple partners? How does that compare against the full length sequence and on and on and on? And so this is where here's a little illustration of that. All right, here's some of the kind of full transporters down here, and you can see that they might be alignable and portions of that type of domain structure exist up here, and maybe they could kind of be corresponding, but it's a challenge. And so this is where we kind of stepped into using some of the new tools that I'm going to talk about today. Cool, we have arrived. All right, made it through that part nice and quick. All right. Okay, so we have this challenge here of aligning these proteins. And at the time, language models, of course, were like emerging as being all the rage, at least in popular media. This was around 2023 when ChatGPT first came out. And we learned that, if you've used ChatGPT, of course, this is a natural language, a spoken language model. You can put in English or other languages and you get English or other languages out. And so this model has been trained on lots of spoken language data. But there are other models that rely on a similar operating principle that are not trained on spoken language data, but on other types of languages like computer languages, like Python or R. And other languages as well, like languages of biology, like DNA and protein sequences. So there are protein language models and DNA language models as well. And so we've used one of these protein language models to try to solve this problem with the ABC transporters. And I want to explain a little bit about the idea of how these language models work, because I think there's interest in that. And I guess maybe you're not in the mood for soup here this time of year, but in Minnesota, we're still in prime soup season. So we're going to do a soup analogy for understanding some of these language models. So imagine this, you have on the left a bunch of soup recipes. And these are relatively distinct from each other, right? Actually, they all make an orange soup, but they may have a different number of ingredients, a different number of steps, different times associated with each step. So at a detailed level, they're different, but they're all kind of vaguely the same thing. All right. Suppose that you have a computer program with many parameters, and parameters here means like knobs that you can tweak. If you're thinking about some big like analog electronic and the soup recipe comes on this side, there's a bunch of knobs that could be tweaked, and what comes out the other side here is, let's say in this example, the abundances of 4,000 different chemicals that would be in the soup. So it's a list of numbers, basically. This is a heat map representing a list of numbers. Here is for the first soup recipe, for the second soup recipe, and for the third soup recipe. All right. So we have this computer program that will translate a soup recipe into some numbers. All right. The next step is this. We then tell the computer program, and I should clarify here, when I say we, that means humans. We're not training these models in my lab or using them, but the people that create these models, this is the process that is generally used. You are at least part of the process. You use the computer program in reverse, and that is to recreate the recipe from the sequence of numbers, or like the nutrition facts, essentially, that were spat out in the first place. And you can imagine that at first this performs very poorly. You probably get some sort of gobbledygook out here. It's going to make some nasty soup, or not soup at all, it's just black tar or something. But we repeat this process many times. We change the settings, so turn all the little knobs and run the soups through again and get numbers and run it backwards and see if we get reasonable soup recipes out. Tweak the little knobs and run the soups again, get the numbers, run the numbers back. Do we recreate the soups? This goes over and over and over again with the knobs being tweaked many times. And if you have enough soup recipes, this actually becomes relatively high fidelity. And actually now it's quite high fidelity. You can decode quite well. And you get a set of numbers that contains essentially the same information encoded in a numerical domain for the soup. You need a lot of soup recipes to do this. So if you had like 650 million soup recipes and 15 billion knobs that could be turned, then this process becomes high fidelity. And this is the concept of what we call an embedding model or an encoder. And people have applied this to proteins. And so the idea is that you input a protein sequence, and it doesn't matter what the length of the sequence is, as long as it's within the limit of the model. It could be short, it could be medium, it could be long. And what comes out is a vector of numbers. And that vector of numbers, importantly, is the same length regardless of the length of the input amino acid sequence that went in. And so if you put an amino acid sequence in, like our ABC transporters, whether it's a full transporter or a half transporter, you get out a number vector that is the same length and could be comparable with the other elements that were put in at the beginning. We're using a set of language models that are part of this family called evolutionary scale. They're pre-trained models, which means that all that knob tweaking I talked about earlier is done ahead of time. So we just deploy these models. This particular one at the time that we used was trained on 650 million protein sequences, 15 billion parameters, although of course there's much more advanced models now. But there's a few perks here. They're available for free. They enable comparisons on sequences that are difficult to align. Relative to multiple sequence alignment, once the model is created, they're much faster. So if you want to add a few sequences in, you can run them through the embedder and suddenly everything is comparable. You don't have to realign everything. The numerical output is quite compatible with machine learning. And this is perhaps the most important point here. They are sensitive to relationships between distant elements in the input. And I'm going to talk about that in detail, but essentially what it means is this. It means that when you've got a linear peptide sequence, over here is the methionine where everything started, and here's the stop codon. That's a linear sequence that the computer sees. But in reality, this is some crazy wrapped up like breathing, moving thing. And well, not breathing in the sense, whatever, it looks like it's breathing. But there could be an amino acid here in the primary sequence and an amino acid here that in 3 dimensions are close to each other and doing something extremely important. And the nature of the relationship between those two is critical for downstream steps. I'll talk more about how this works when we talk about natural language or spoken language in a second. But the idea here is that these models are sensitive to relationships between distant elements in the input. They're not simply encoding, oh, the first amino acid is methionine, so we get a 3 here. The second amino acid is the threonine, so we get a -1 here. This 3 represents a essentially the first, well, it represents a bunch of things. It's all like intertwined. And one of the parts that's intertwined is relationships between amino acids that are distant. Okay. Hello? There we are. So here, this is a two-dimensional space. This is like a UMAP space. It's sort of like a PCA. If you haven't heard of UMAP, it's a little bit different, but more or less the same. A two-dimensional representation of those numerical vectors that were derived from our ABC transporters. And here, the ones that I'm showing first, this is a list of previously, this is like the control group. This is the list of previously characterized ABC transporters from plants that we collected that have known substrates. And they're grouped here according to kind of generally speaking the family that they belong to. So you can see that there's a few different families, and we can see some of their substrates. So a few of these proteins down here were characterized in Arabidopsis by other groups and shown to transport several different wax compounds. Importantly for our conversation here, it's these long chain-like compounds. A few of these others transport cutin precursors. If you're familiar with cutin, some of these down here in this little subgroup are transporting things that have rings in their structures. This is interesting to us. Remember, our triterpenoids have rings in them. So maybe we're looking for something that's down in this group, perhaps. Auxin transporters are a major group of ABC transporters, excuse me, and they're located up here, for example. So I'm not going to go maybe too much into detail, but we used these numerical representation of the proteins to generate some hypotheses about which transporters might be most relevant to triterpenoids. And we combined that information with expression data to identify a number of top candidates that we wanted to test. Yeah, and then we tested them. So Taylor, a master's student in my lab, did the tests. You're all probably, well, no, that's not true. If you're not familiar with the system, the way it works is you put a gene into an expression vector, you put it into Agrobacterium. This is a plant pathogen. The pathogen infects the plant and causes the plant to express the gene of interest. And then we can analyze the surface chemistry of the plant with gas chromatography mass spectrometry. So Taylor took this on when she expressed when she infected the plant with Agrobacterium alone or with green fluorescent protein, the amount of, this is like the relative amount of triterpenoid on the surface. We see essentially nothing. If she expresses this gene here, it's called GGBAS. This is a triterpenoid synthesis gene. So to create the substrate that we anticipate being transported, she sees maybe like a little squeak of triterpenoid on the surface in some cases, although we think this is coming from cracks in the leaf. But anyway, if she expresses any of the transporters alone without the gene that generates the triterpenoid substrate, we also see nothing on the surface as far as triterpenoids are concerned. And for some of the transporters, if she expresses them in combination with the synthesis gene that makes the substrate, still nothing. But of course, some of them, it wouldn't be a very good story if I had nothing here. Some of them, when we express those transporters in combination with the substrate generation protein, then we see higher levels of triterpenoid accumulate on the surface. And we have some follow-up experiments. Of course, we wanted to know if these transporters were specific for triterpenoids versus having a broad specificity. And it turns out that these ones that we found in Kalanchoe are actually relatively broad. They're a wax transporter that transports both kind of traditional chain-like molecules in addition to triterpenoids, which is a new finding, albeit relatively niche. But anyway, what helped us here to narrow down our candidates was that use of the protein language model. All right, I want to summarize quick and then move on to a bit more language model work. Oh, actually, no, we're not at the summary. Okay, sorry. But maybe this is, we can pick up right here. So I began that previous section by all that business with triterpenoids and so forth. I began it by explaining how we got there was looking at the distribution of different chemicals across the plant phylogeny. And that's what led us to, oh, look, this particular group here, these succulents, these Kalanchoe plants, they have a funny chemistry that isn't found in any of the other species we looked at. And then we used that information to find a transporter with unique characteristics. This illustrates a workflow that could be could be used for lots of different things. Sorry, my watch makes the same noise. This workflow could be used for lots of different types of discoveries, right? Looking at the distribution of some trait across a phylogeny. In fact, like this is nothing new, right? This is like the basis for like QTL populations and so forth. Anyway, or QTL mapping. But anyway, the idea is that if we look at the distribution of chemicals across a phylogeny and find unique kind of characteristics, we can dig down deeper and find some interesting underlying biochemistry. And there have been multiple examples of that. I just showed you one here with Kalankoe. There's a really famous and beautiful example that I like from Hiroshi Maeda's group in Wisconsin-Madison, where they found a variant tyrosine synthesis enzyme. And this is what has enabled a variety of specialized metabolites to evolve, including like, beets that are so red. That red is coming from a unique tyrosine metabolism that came from this lineage-specific tyrosine adjacent enzyme. Anyway, Hiroshi and I were talking about this at a meeting, and we thought, well, there's all kinds of different data that we could look at in a phylogenetic context. We could look at enzyme activities, plant-insect interaction, plant chemical occurrence, like I showed you here, or whatever it might be. And we could learn all kinds of things about evolution and biochemistry and so forth. But the problem is that we need lots of data to do this. We need lots of data to map onto the phylogeny. And how do we get it? All right, fortunately, we have many data points, right? Scientists have been working on plants and of course other organisms for a very long time and generating what we can think of as labeled data. And in the example I'm going to talk about today, that would be like, oh, this particular plant species makes this particular chemical according to this article where measurements were made. There's a piece of labeled data. These data points are reported in literature, but they're not always organized systematically. And there are databases of these, of course, but it's impossible to keep up with organizing them, and they're not comprehensive, and maybe they're not including some of the sort of unusual plant species that might be most interesting to us. So one solution here might be to read many papers and tabulate data manually, and we have done a lot of this, but it's slow. And so we wondered whether a potential alternative solution would be to use a language model to extract this data from text and organize it in a systematic way. And we wondered whether that could be done accurately. And so this is a project that brings us to our sort of third section here. And in order to talk about this, I want to go a little bit further beyond what we talked about with the soup analogy. The point of the soup analogy was to illustrate that the transformer-based language models, one of the key underlying principles is that it takes some input, and I talked about protein sequences, but it could be images, it could be text, it could be whatever, and it transforms that input into some set of numbers that is of a constant length, regardless of the input. And importantly, those numbers can be decoded back into the original input. And so the information that's in the input is represented in the numbers. Okay. That's what's happening here. So we've got the word sea, like the ocean, two related words. This set of colored blocks here is representing a heat map of the numbers, as you can see on the scale here, that would represent the word sea. If you're curious about this, the QR code here leads to the article where this is from in the Financial Times. It's quite interesting. But the idea is that these two words have similar meanings and their numerical representations are similar. That makes sense. Same for football and soccer. Same for I and we. But what's important here in the context of this natural language processing, and as I alluded to with the proteins when talking about distant elements in the sequence that might be close together in 3D and therefore important, that relationship that is defined over distance can be captured by these models, and this is how. So here we have a sentence, the bank's interest rate rises. Okay, there's the word interest here. And so based on what I just showed you on the previous slide, what I showed you on this slide here about sea and ocean and football and soccer and whatever, that word interest is going to get some type of numerical vector representing it. So this word interest will get a numerical vector. But the next step, and the important step, is that vector of numbers, that list of numbers, it is modified slightly according to the words that occur around it. And so that numerical representation contains not only the information of, oh, this numerical, this list of numbers means interest, it means interest in the context of bank and rate and rising. This is the financial meaning of the word. We could take this other sentence down here, or excuse me, let's have a look at these sentences quick, yeah? So here's this interest that has the financial sense, rising, rate, bank. And so the vector representing this word here is going to be modified by those neighbors, whereas this one here, which is the same word, and so its initial numerical representation would be the same as the first one, But at the end of this sort of secondary process that I'm describing, which is called the attention mechanism, in case you've heard that word in this context, the vector representing this word is modified by these words more strongly. Know, in, hearing, I. This meaning of the word interest is much more so to do with like whether or not a person is interested in something, whether they want something, right? It's a different meaning of the same word. And so via this mechanism that modifies the numbers representing a word according to its context, according to its environment and what's around it, this is quite powerful because it means we can now distinguish between these two things. And you could imagine in the protein world, this could be powerful, right? You could have a particular amino acid, and the numerical representation does not just mean, oh, this is a glutamate. It means it is a glutamate in this particular environment. And it could be distinguished from a glutamate that is in the same position in the multiple sequence alignment. So in multiple sequence alignment space, it's indistinguishable. But the vector that encodes it is distinct because that glutamate resides in a distinct environment. In our context here, this type of attention mechanism, paying attention to the words next to you, it means that there is some understanding in this model of the context around the words that it is seeing. And it can use that to generate some kind of output. And so these models can do a variety of tasks. We've talked about embedding models, which could, in this case, convert words into numbers. But there are more complex things, like if you've used ChatGPT or Claude or Gemini or any of these, it's like a chat model. So you put in some question and you get an answer out. So it's a generative model. You can also get these models to do classification, to label something according to a set of predefined categories. Or there are multimodal ones, as I mentioned, that can take images or other sorts of video input and so forth. But we're interested in this one here, right? Maybe I'll take a step back and remind you that this whole business I'm talking about is mapping chemicals onto a plant tree of life. We want to know where those different chemicals occur. And I told you that there are lots of articles that say in their abstract, oh, we studied this particular plant and found chemical X and chemical Y and chemical Z using analyses that involve liquid chromatography, mass spectrometry. And so what we want to do, what we're trying to figure out is if we're going to map phytochemicals onto phylogenies, we need to be able to figure out, can the language model look at that abstract and accurately determine that that particular plant makes that chemical? And importantly, not just do those words co-occur in the same text. That would just be a pattern matching. That's not what we need. It might have said, we looked at this plant and did not find this particular chemical. So co-occurrence doesn't work here. We need something that understands a bit about context in order to provide more accurate outputs. And so our hope was that this mechanism would be a way of doing that. So we generated like a ground truth or labeled data set first. We grabbed a lot of references from SciFinder and the list of plant species from World Flora Online, and we essentially manually evaluated a wide array of abstracts and did the task that I just mentioned. We asked, which plants are making which chemicals and made a big list. Okay, so we worked with Hiroshi's group to do this. We looked at over 2,500. Actually, these were full texts that we looked at. And we populated kind of this map here. This is a summary of the map. It's quite a bit richer. But here we've got many different plant orders and many different chemical compounds here. And you can see that, for example, this group of compounds is only produced in this particular lineage, or this group of compounds here is only found in that lineage, versus some of the ones at the top that are more broadly distributed. So we had this data set with which to work. And a student in my lab, we were using OpenAI models at the time, though now we use Anthropic, of course. But we took our data set and we began to pass the rows. And the rows were an abstract, the name of a plant species and the name of a plant chemical. We pass that row to the language model and ask, does that particular species make that compound according to what's described in the abstract? And you could imagine many different ways of asking that question. Do they provide empirical evidence for that association, or do they simply mention them together or whatever? So there's a bunch of different ways to ask this. But we essentially measure the accuracy of the responses against our ground truth data set. And there are a variety of possible responses, of course. I'll go through them briefly. So this is quite a long time ago. These models are not even available anymore. Quite a long time ago, being like a year and a half ago. Well, let's see here. We can look at that. Let's work with some of the easier ones first. So true positives. This is where the model correctly identifies what we also identified as a correct association between a particular plant and a particular chemical. So we got the answer. The answer was yes, and it got it right. There are some others that are interesting. True negatives here. This is where the words simply co-occur, but there is no actual evidence stated in the abstract that that plant actually makes that chemical. And the model got it correct. The model said, yeah, these co-occur, but there's no evidence that this plant is actually making this species. False positives are where it erroneously attributes a chemical to a species, and a false negative is where there is a true association there, but the model doesn't recognize it. The pattern that we should look at here is this one. These models are ordered from smallest to largest, and what small and large means here is the number of parameters. It's the number of little knobs on that analog device I was talking about that would convert soup into numbers. So the more parameters that the model has, it sort of appears to be, at the risk of anthropomorphizing, a bit more skeptical in that it misses many more true associations, but it has a pretty low false positive rate. So if it says, yes, this particular plant makes this chemical, that's probably correct. It has a relatively low false positive rate, but it misses lots of correct answers in trying to be stringent. So it seems like the size of the model matters, and we became interested in what other kinds of optimizations could be done. Let's see here. We have 10 minutes left. I want to make sure I get this right. We tried a variety of things. I mentioned earlier that depending on how you ask the question, it can make a difference. So we created 50 different, was it 50? Yeah, 50 different ways of asking the model to do this task. And these are all different phrasings, right? You could say, look at the abstract and tell me, does the plant make the chemical based on evidence in the abstract? Or you could say, I'm going to show you an abstract and I'm going to ask you about a chemical. Here's the chemical and here's the plant. Are they associated? Or there's a zillion different phrasings to ask the same thing. And so we tried many different ones. A lot of them had about an 80% correct response rate, but some did better and some did worse. What was interesting, so we took all those prompts and embedded them. We made their numerical representations and looked at them in a PCA space. And we can see that the poor performing prompts, which are shown in red here, they kind of cluster together, meaning that linguistically they are related. And the prompts that performed well also kind of clustered together. And there's a statistical significance here. It's not a huge effect size, but it's detectable between these two different clusters. And so the prompts that did well also had some linguistic similarities in terms of the order of the words and their phrasing. And what we think this means is that when we ask, this is called prompt engineering, so when we ask a model to do a task, the prompt specifics on the word level can matter, even though that seems like a strange thing. We tried a variety of other things. I want to make sure there's time here. So we tried retrieving excerpts from literature. This is the important result here. There's this, let's see, we'll make it quick. There is an aspect of prompt engineering that is called role-based prompting, and it's where you give the model a role. So we told it, for example, you are an AI classifier. Classify this connection between this chemical and this plant species as true or false based on the abstract, or something like that. But telling it that it was an AI classifier, as opposed to just asking for the task outright, led to a measurable difference in performance. And we tried various other things that had other effects, but this role seems to be quite important. And this, I think, is connected to how the training of these models works. I want to briefly talk about the environmental impacts of language models, because this is something that I think we hear a lot about. And so it's an important topic. And the way that I want to do this, of course, is maybe to provide some kind of broader context and some kind of numerical data to think about environmental impacts from sort of a systems thinking point of view. For that, we're going to go here. So I've made this little app. Here we're looking at CO2 emissions, approximate emissions from various activities. And maybe I can zoom in here a bit. Yeah. Okay, so here's 2 minutes of microwave use. Maybe this emits around, I don't know, 8/1000ths of a kilogram, okay? 8 grams of CO2. And we can add a few things to this chart. So here's maybe something like 100 AI queries, which if you're using an agentic AI system, maybe you're doing, well, maybe not 100 in one go, but maybe a dozen. We're looking at 30 grams of carbon. Here's some other activities. Streaming Netflix for an hour of laptop use, a latte. I have one of these every morning. And of course, this is accounting for the production of the milk and the coffee and so forth, right? That's why this number is high. Using a space heater. I use one in my basement. Driving a car for a mile. A 5-minute hot shower. Clothes dryer cycle, or eating a hamburger. My point here is that an individual's use, or maybe even a research lab scale use of AI is comparable to other things in our daily lives. Okay. And the same is sort of true for water here. Okay, here's a cup of tap water. This is the water perhaps used at the data center approximately for 100 AI queries, a toilet flush, a hot shower, an apple, coffee, milk, again, a hamburger. Again, comparable to other things that we do in our daily lives. I'm not trying to minimize the environmental impacts of AI, and in fact, we'll talk about it at scale in a second. My point here is that if a person's goal is to reduce their carbon footprint, there are a wide variety of ways that might be more impactful than abstaining from AI use. I'm not saying that you shouldn't also abstain if that is what choice you would like to make. I'm simply saying that by reducing the amount of flying you do and by eating less meat, you would have a much bigger impact. Okay, but why do people worry about AI in the context of the environment? And it's because of industrial scale. People are thinking about automation of entire factories with AI-powered robots, or entire financial and legal enterprises built on these types of systems that are running constantly at large scales. And at those scales, the impacts are real, right? Data center emissions are currently about 1/5 of the global aviation emissions. All right, the sources are down here in case you're curious. And of course, large data centers use millions of gallons of water comparable to a university campus or a small town. So this is no small usage at larger scales, but this is like a drop in the bucket kind of situation. Maybe what it means is if you're concerned about this sort of thing, a way that you might go about it would be to pursue careers in places that are going to have positive impacts with regard to this technology. Sorry, this slide was made for a chemistry class of mine. So of course I've got battery chemistry and energy storage here, but biomaterials and biotechnology are clearly relevant to this audience. And energy environmental use policy may also be. In the wake of large language models that are also called frontier models, those are the ones that are most closely related to the data I just presented in that scale that I talked about. In the wake of that development, there's also all kinds of other models being developed. And this is maybe the most exciting thing, in my opinion, that's happening now. And these are called small language models. And the reason that I have the word local here is because you can run them on your laptop if you would like. These are generally, this is sort of the hub for this whole movement. It's called Hugging Face. If you haven't been to Hugging Face, they have tutorials as well as lots of models that you can download that have been kind of optimized for specific purposes. And these have much smaller impacts, and they're sort of comparable, running them even at sort of scale is comparable to like a Netflix stream or running a web search or something. And so of course we wanted to know, in our task of identifying connections between plant chemicals and plant species, can these small models do the job? Can they perform as well as the models that I presented to you earlier? This might be a less impactful way to do our research. So I just have a couple minutes left, so I'm going to go through this a bit quickly, but maybe here's the take-home message. Here's a particular model. This one's called Quen 2.5. It has 32 billion parameters. It has instruction fine-tuning, so it follows instructions well. And here in green at the top, these are the true negatives. It is if it correctly recognizes that there is not an association. True positives here in green, correctly recognizing a true association. In red are when it gets something wrong, either calling a true association, not recognizing a true association, or making up the fact that there is an association when in reality there's not. So for this model here, you can see 26% and 67%. The sum of those is how many it got correct. We tried a wide variety of models of different sizes and different speeds from different labs and so on. You can see that generally speaking, they're performing quite close. This line, this horizontal line at the top here, that is like what the correct answer is. So there's like a slight imbalance in positives versus negatives in the data set. But you can see that by and large, many of the models do quite well. The take home here is that for this particular task, the large models and the small models both had around 90% accuracy in their reporting. And with the small models, they're not only kind of accurate, they're reasonably accessible and they're democratized. You can edit these, you can give them labeled data and fine tune them to give you outputs that are more similar to your examples. It keeps your data local. So if you're working with sensitive data or unpublished data, or whatever it might be, nothing leaves your machine. Also, the environmental impacts are, of course, smaller. Right. All right, so that kind of covers the three areas here. I hope that was a reasonable trajectory. We talked about community science leading to this data set that looked at wax chemistry. We followed the wax chemical leads to identify an interesting biochemical phenomenon that we followed up on. And now we're working on not only understanding stronger relationships between protein structure and function using language models, but using the natural language processing, or excuse me, spoken language processing models to begin to scale this approach and generate other avenues for discovery. Well, this is what it looks like in Duluth in the fall. Just on the other side of the hill there. Okay, all right, so thanks to the folks here. This is the team that worked on Kalankoe, our language model team, and the sorghum model team, although the lineup has shifted a bit, but thank you to the USDA and the RCSA for funding, and we have a PRF project starting in the fall. And thank you for your attention, and I'll take your questions.
Oh yeah, for your ESM work, you said you use 15 billion models. I think that's ESM2. Did you do any like fine tuning before you plugged into the UMAP, or did you just use the raw pre-trained ESM?
For what I showed today, we just use raw pre-trained ESM2. We're now using ESMC, using
cool.
residue level embeddings and not like summarized embeddings. But for what I showed there, that was ESM just without any sort of fine tuning.
Can I ask you one question about ESMC usage? So like there's like multi-track inputs. Do you
ESM3, yes.
Yeah. Okay, you're 100% right. Never mind. You're good.
Really interesting work. I enjoyed that a lot. So this is a naive question because I don't know anything about these AI models. But when you were showing the example of sea and ocean, for example, so C has 3 letters, and it was represented by this long string of numbers. And I guess that the answer is that C can exist in different contexts. I guess I wanted to understand the relationship between the representation of 3 English letters with however many numbers you had in that string. And does that change with the complexity of the model, and how is that mapped?
Yeah, this is a good question. And maybe I'll say, as far as this might be a naive question, was kind of how you prefaced it. One of my favorite parts, unless a person is in computer science or bioinformatics where they've been working on these things for years, we're all kind of new to this stuff and we're all kind of learning together. So it's not a silly question. Sorry. Okay. Actually, I think this question might be most easily answered by considering an amino acid example. My understanding is that the way these models are trained is that they have what's called a context window, essentially. And so there is some like maximal length of input that they will accept. And for amino acids, like let's say you give it just a 4 amino acid peptide, and which would be like similar to C, like what you're talking about. And then we're gonna ask it to generate the numbers. And I mentioned that regardless of the length of the input, as long as it's below that maximum size, the length of the number output will be the same. And what it does is it adds padding. And so it would add, like if the window was 1000 and you gave it a 4 amino acid peptide, it would add 996 like little representations of like, there is nothing here. And then that full length input would be passed and encoded. And when it's like, if you were to decode it, you would get that same representation out. So you would get your 4 amino acids with a bunch of like, kind of like spaces appended. The second part of your question was, how does this change as there are different parameters and approaching different models? And yes, it does change. Different models give different lengths of outputs. Some are very large, which means they're like have the capacity potentially to encode a very complex input. Some might have like a short set of numbers coming out. Maybe like a small one would be like 256 numbers, and so maybe the representation is simpler and maybe is not as well suited to encoding like something quite so complex, but maybe would work better at scale, or depending on like what the downstream task was, might be suitable. Does that answer you?
I'm gonna meet with you later, so
Oh,
I'll wait.
So do you train any chemical language model with the phytochemical texts?
So we have not trained a language model with the phytochemical texts, but we have done what's called fine tuning. It is where I think the way to think about it maybe is, I was talking about this like analog board with all these knobs on it, and this is the model with many parameters. Each knob is a parameter, and we have an input going in and a bunch of numbers coming out. And in the case we're talking about, there's the input is an abstract and a question about this plant and this species or whatever. We have been using pre-trained models because the scale of data needed to train a model is very large. But what can be done, and what actually can be done with quite a small number of examples, is what's called fine tuning. You could imagine taking like 1% of all those knobs and putting like a coefficient next to each one. That maybe, let's just say it ranges from 0 to 1. Well, sure, 0 to 1, a coefficient. And maybe what we'll do is, rather than train the whole model, we'll take our example, our labeled data where we know what the answer is, and maybe we've got 100 of those, let's say, and we'll run this routine where we pass that through, and we know what the expected answer is. But if the model doesn't get it quite right, we'll change some of those coefficients. And it's just 1% of the parameters that have coefficients, but we'll change those so that what comes out after the coefficients are applied is more faithful to what we expect the answer to be. And we can do this with our 100 examples to kind of get the coefficients as well tuned as possible. And this can lead to substantial increases in the performance of a model. In our case, we did that for one of the Quen models. And it's interesting. It didn't really change the ability of the model to recognize put out true positives, to correctly recognize a true association, but it greatly increased the ability of the model to recognize negative results, to recognize when there was not an association there. We made sure that the negative results were like included in that fine-tuning set.
Dr. Monin is asking a question on Zoom.
Yeah, Lucas, thank you very much. Very interesting talk. I've got, maybe it's a bit.
Yeah, I'm sorry, Dr.
No,
I can't
we'll have to
hear
do
you,
it
but-
through here. Yeah.
Can you not hear me?
Robin's here. And
Yeah,
she's
I'm
helping.
trying my best, Deb, okay?
Should I talk? Hello?
Maybe you can put it in the chat. Yeah.
Okay, I'll put it in the chat. Thank you.
She'll put it in the chat. We'll check in a second.
Yeah, because we can't hear you, Deb. There's no sound coming back. You have to put it in the chat.
I think she's muted.
No, I've got my speakers now.
There she goes. A philosophical
You can tell it.
question.
Deb, we can hear you now if you want to unmute.
You can hear me now?
Yeah.
Oh, yes. Okay. So a bit of a philosophical question. Great talk.
Please
I
keep
learned
going.
a lot. I learned a lot. You clearly had to put a lot of energy into testing and working with the AI to determine whether the answers you get are reasonable or not. You started off the talk with some questions about secondary metabolites. So my question to you, and I do have a concern about this as we train the younger scientists, is we now have a method that can be incredibly powerful. It takes a lot of time to figure out how powerful, and that sucks time from thinking deeply about the biological questions. So you're right in the midst of this right now, and you're training young people. So how would you recommend going forward, especially training the young people, to consider using what AI has to offer versus one, getting trained in depth in the background? And you'd argue, well, maybe you can get that from AI by doing these searches, but it takes a lot of work to get that background. And then getting to the point of taking the time that's necessary to ask the deep biological questions.
Question,
Yeah,
because
absolutely.
the audience online didn't
Okay,
hear.
thanks, Deb. Okay, if I'm understanding correctly, the question sort of has a few different parts. You began by saying like lots of work needed to go into validating that things were working correctly. Some concerns. One concern is essentially a balance of time between spending time learning like disciplinary or gaining disciplinary expertise and asking and thinking about deep biological questions versus figuring out these tools. And then later on, sort of a balance of like, how should we think about like those two things? What is there a danger in sacrificing disciplinary expertise or giving up some of the role of disciplinary expertise to AI? Did I get your question correct, Deb, before I answer? Yeah, okay.
Yes.
So yeah, this is something I think about a lot, and I think about it for myself, and we actually should all be thinking about this. In my current thinking sort of has maybe the simplest answer is this. Maybe I'll give like the peer-reviewed answer. So people have studied this, and the answer that is emerging is that it is the combination of disciplinary expertise and the ability to use powerful tools that lead to the best outcomes. And that is in part because disciplinary expertise is required in order to validate and have a sense, any type of intuition about what the contributions of an advanced technology might be to a research project. And without the disciplinary expertise, there is essentially no ability to either ask the right questions, to know if what has been produced by the research project is meaningful, and in addition to I find to communicate about it. Without disciplinary expertise, standing up and giving a talk like this is terrifying, because the questions that come inevitably have to do with the biological or biochemical knowledge we're trying to apply this technology to those things. And so without the disciplinary foundation, there isn't any foundation to kind of build upon. And so my suggestion, part of your question was like, we're working with folks that, like yourselves, like all of us, as I just said earlier, that are learning how to do this. My suggestion is a parallel approach. As with any new tool, we need to learn what it can and can't do, which requires experimentation. But in how to apply that requires disciplinary expertise. And these two things go hand in hand in my mind. I hope, did that address your question? Thank you.
All right.
Yeah, thank you.
Yeah, one last question.
You said ESM compared to multiple sequence alignment is faster and useful. It raises a question for me. Does it matter if you insert with the aligned sequence or not aligned sequence into ESM? Because that's the that you can compare them or not.
This is a good question. This is something that we spend a lot of time on. So there are some variants of ESM that have been trained to accept multiple sequence alignments, and so they behave in, I think, more interpretable ways when an alignment is passed. We haven't used those as much, but I know they exist. The base model is trained on raw peptide sequences going in. That's all I should be saying. Yep.
All right, well, let's thank Luke again.